The ability to contract and movement are basic properties of all animal cells. Muscle tissues are the best example of these properties. Muscle tissue is a soft tissue that composes in animal bodies and gives muscle the ability to contract. It is formed during embryonic development through a process known as myogenesis. We have around 650 muscles in our body, which make up roughly half of our body weight. All muscles can stretch and contract very rapidly and reversibly. By doing so, they exert a mechanical force on their neighboring cells and tissues. Muscles are of various types according to the functions they perform, like maintenance of body posture and orientation, circulatory and gastrointestinal movements, and various other functions required by the organism. The functional unit of a muscle is a muscle fiber. Many of these muscle fibers fuse to constitute a whole muscle. Some of the muscle fibers are small and short, while others have large muscle cells running the whole length of the muscle. Muscle fibers are covered by a layer of collagen fibers and connective tissue called the epimysium. The epimysium protects the muscle from friction against other muscles and bones. It continues to the end of the muscle to form muscle tendons along with the other connective tissues. The cross section of a muscle shows bundles of fibers known as fascicule, surrounded by another connective tissue called as the perimysium. Each fascicule contains 10 to 100 muscle fibers. Most of the structure of muscle fiber is made of myofibrils and myofilaments, which are the contractile structures of the cells. Myofibrils are made of many protein fibers arranged into repeating units called sarcomeres. The sarcomere is the functional unit of the muscle fiber. On the other hand, the sarcolemma is the cell membrane of muscle fiber, which is a typical plasma membrane and acts as a conductor for electrochemical signals that stimulate muscle cells. It lies beneath the epimysium. The fluid portion of the muscle fiber is called as sarcoplasm. This is the cell cytoplasm and is a gelatinous fluid filling most of the cell. It contains glycogen and fats for energy along with the organelles. Sarcoplasmic reticulum serves as a storage for the calcium ion necessary for muscle contraction. A muscle fiber is rich in sarcosomes, that is mitochondria, that provide energy in the form of ATP for muscle contraction. Sarcomeres are made of two types of protein fibers, thick filaments and thin filaments. Thick filaments. Thick filaments are made of many bonded units of the protein myosin. Myosin is the protein that causes the muscles to contract. Thin filaments. The thin filaments are made of three proteins. Actin. Actin forms a helical structure that makes the majority of thin filaments. It forms myosin binding sites that allow myosin to connect to and move actin during muscle contraction. Tropomyosin. Tropomyosin is a long protein fiber that wraps around actin and covers the myosin binding sites on actin. Troponin. Troponin is bound very tightly to tropomyosin and moves it away from myosin binding sites during muscle contraction. There are three different types of muscle tissue depending on function and location in the body. In mammals, these are skeletal or striated muscles, smooth or non-striated muscles, and cardiac muscles, which are sometimes known as semi-striated. Smooth and cardiac muscles contract involuntarily without conscious intervention. Striated or skeletal muscles only contract voluntarily upon influence of the central nervous system. Muscles respond to various neurotransmitters and endocrine substances such as acetylcholine, noradrenaline, adrenaline and nitric acid. The response of different muscles to these substances depends on muscle type and the exact location of the muscle. Skeletal muscle is made up of individual muscle cells or myocytes, known as muscle fibers. They are formed from the fusion of developmental myoblasts in a process known as myogenesis. Myoblasts are a type of embryonic progenitor cells that give rise to a muscle cell. The muscle fibers are very large, multinucleated and up to several millimeters in length. Skeletal muscles are attached to the bones and have the main function of contracting to facilitate movements of our skeleton. They appear striated due to the presence of the proteins actin and myosin, because of which they are also known as striated muscles. These proteins form the sarcomere found within the myofibrils. Skeletal muscles are also called as voluntary muscles because we can control them directly. This is through the nerve impulses from our brain reaching the muscles. These contractions can produce powerful and fast movements or small precise actions. Skeletal muscles can also return to their original shape after stretching or contracting. Like a typical muscle cell, a skeletal muscle cell contains sarcoplasm, sarcolemma, mitochondria, myofibrils and sarcoplasmic reticulum. At regular intervals, it has dilated end sacs known as terminal cisternae. These cross the muscle fiber from one side to the other. In between two terminal cisternae is a tubular infolding called a transverse tubule or T-tubule. T-tubules are the pathways for action potentials that signal the sarcoplasmic reticulum to release calcium, causing a muscle contraction. Together, two terminal cisternae and a transverse tubule form a triad. As a T-tubule passes each myofibril, it touches the sarcoplasmic reticulum. As the action potential in a T-tubule reaches each piece of sarcoplasmic reticulum, the calcium channels in the sarcoplasmic reticulum open. As a result, calcium flows out of the sarcoplasmic reticulum and into the sarcomere with its thick and thin filaments. This causes the filaments to start sliding and the sarcomere to shorten. But very quickly, the calcium is actively transported back into the sarcoplasmic reticulum and the sarcomere relaxes. The thick filaments are comprised of myosin. Each myosin molecule is shaped like a golf head. The head points out from the surface of the thick filament, which forms a structure called as the cross bridge that binds to the thin filament made of actin. Within skeletal muscles, there are three types of fibers depending on speed of contraction, different types of activity and variation in color. The color variation is due to the myoglobin content, which is the oxygen carrying protein present in muscles. The three types of muscle fibers are type 1, type 2A and type 2B. Type 1. Type 1 fibers are also known as slow twitch fibers. They are red in color due to the presence of large volumes of myoglobin, so that the oxygen carrying capacity of the muscle is very high. For production of ATP, high numbers of mitochondria are present. Due to this, the muscles which have type 1 fibers have high endurance capacity, example postural muscles in neck and spine. This also makes skeletal muscles very resistant to fatigue, so that they can undergo repeated low level contractions. Athletes such as marathon runners have high number of these fibers, partly through genetics and partly through training. Type 2A. Type 2A fibers are also known as fast oxidative fibers. They are a hybrid of type 1 and type 2 fibers. These fibers contain a large number of mitochondria and myoglobin, because of which they are red in color. They use aerobic and anaerobic metabolism to produce and split ATP at a fast rate. This is the reason why they can produce fast and strong muscle contractions. However, they are more prone to fatigue than type 1 fibers. They are a part of the leg muscles and are used during prolonged running. Type 2B. Type 2B fibers are often called as glycolytic fibers and are white in color due to low level of myoglobin. They also contain few mitochondria. They produce ATP at a slower rate by anaerobic metabolism and break it down very quickly. This results in short, fast bursts of power and rapid fatigue. They are found in large quantities in the muscles of the arms. Skeletal muscles perform two functions: support and movement, and homeostasis. Support and movement. Skeletal muscles move the body. Skeletal muscle contractions pull on tendons which are attached to bones. If contraction of the muscle causes the muscle to shorten, the bone will move, causing the body part to move. However, the contraction of skeletal muscle does not always result in the movement of body part. This is the case during maintenance of body posture and position. For example, holding your head still while you are watching this lesson. The muscles in our abdominal wall and pelvis contract to maintain our abdominal organs in position. A special type of skeletal muscle called a sphincter regulates the opening of our digestive and urinary systems. A sphincter is an organ composed of muscle that surrounds a hollow organ. Example, intestines and blood vessels are hollow organs. The sphincters at the end of our digestive and urinary systems are composed of skeletal muscle and provide conscious control of swallowing, defecation and urination. Homeostasis. In addition to providing support and movement, skeletal muscles are necessary for some homeostatic functions. Homeostasis is the maintenance of a relatively constant internal environment, especially relating to temperature, that is, temperature regulation. As energy is used to contract muscle, much of that energy is converted to heat, and heat helps to maintain a relatively constant body temperature. Since much of our body is skeletal muscle, these organs provide an effective means of temperature regulation. Shivering thermogenesis is a rapid contraction of our muscles for the purpose of generating heat. Therefore, skeletal muscle helps with homeostatic regulation of body temperature. Smooth muscles are also known as involuntary muscles as we cannot control its movements. They can also be called as unstriated as they are not striped like skeletal muscles. They are found in the walls of hollow organs such as stomach, esophagus, bronchi, and in the walls of blood vessels. Smooth muscle fibers are much smaller, 2 to 10 millimeters in diameter, than skeletal muscle fibers, 10 to 100 millimeter. It is divided into two subgroups, the single unit or unitary and multi-unit smooth muscle. Within single unit cells, the whole bundle or sheets contracts as a syncytium, that is, a multinucleate mass of cytoplasm that is not separated into cells. Multi-unit smooth muscle tissues innervate individual cells. They allow for fine control and gradual responses, just like motor unit in skeletal muscles. Smooth muscle is found within the walls of blood vessels called as vascular smooth muscle, lymphatic vessels, urinary bladder, uterus termed uterine smooth muscle, gastrointestinal tract, respiratory tract, arrector pili of skin, the ciliary muscle, iris of the eye, and glomeruli of the kidneys called as mesangial cells. Most of the volume of the cytoplasm of smooth muscle cells has myosin and actin. Together, they make the entire smooth muscle tissue contract with them. Myosin is primarily of class 2 in smooth muscles. Actin is made of alpha and gamma actin, where alpha actin predominates. The ratio of actin to myosin is between 2:1 and 10:1 in smooth muscles, compared to approximately 6:1 in skeletal muscle and 4:1 in cardiac muscle. Smooth muscles do not contain the protein troponin. Instead, calmodulin, which has a regulatory role in smooth muscle, caldesmon and calponin are significant proteins expressed within smooth muscle. Calponin molecules have been proposed to be a load-bearing protein, and caldesmon enhances the ability of smooth muscle to maintain tension. The myosin and actin are the contractile parts of continuous chains of tensile structures that stretch both across and between smooth muscle cells. There are tensile structures present in smooth muscles. The actin filaments of contractile units are attached to dense bodies rich in alpha-actinin. These dense bodies also attach intermediate filaments and serve as anchors, from which the thin filaments can exert force. The intermediate filaments consist of vimentin and desmin. Dense bodies also are associated with beta-actin, which is the type found in the cytoskeleton. The intermediate filaments are connected to another intermediate filaments via dense bodies, which eventually attach to adherence junction, also called focal adhesions, in the sarcolemma of the smooth muscle cell. As a result, cells are mechanically coupled to one another such that contraction of one cell causes some degree of contraction in an adjoining cell. Gap junctions couple adjacent cells chemically and electrically, facilitating the spread of chemicals, example calcium, or action potentials between smooth muscle cells. The dense band areas alternate with regions of membrane containing numerous caveoli. When complexes of actin and myosin contract, force is transduced to the sarcolemma through intermediate filaments attached to the dense bands. Caveoli are microdomains of lipid rafts that are only specialized to cell signaling events and ion channels. They are close to sarcoplasmic reticulum or mitochondria and organize signaling molecules in the membrane. A smooth muscle is excited by external stimuli which causes contraction. For example, blood vessels in skin, gastrointestinal system, kidney and brain respond to norepinephrine and epinephrine by producing vasoconstriction. The smooth muscles have several important functions. The most important is the ability to compress, contract or extend ducts, organs, tubes and sacs. They are found primarily in the heart, digestive tract and the stomach. The stomach contains a circular smooth layer inside and the longitudinal smooth layer outside. These two layers help in the proper digestion and metabolism of ingested foodstuffs taken in by the body. Smooth muscles play an important role in the functioning of the heart. Without the smooth muscles, the heart will not be able to contract, relax and pump blood to the rest of the body. Due to the elasticity of the smooth muscles, it is an important component of the kidneys, lungs and blood vessels. These body parts need to be elastic to perform their functions like increase in the size of urinary bladder according to urine input or expansion of lung after inhalation. The structural and functional characteristics of heart muscles are intermediate between those of skeletal and smooth muscles. The heart muscles are striated and contractions are strong and utilize a lot of energy like skeletal muscles. Like some smooth muscles, cardiac muscles can contract without nervous stimulation, that is, it is myogenic and the contractions are not voluntary. They are controlled by autonomous nervous system. Muscles in different parts of the heart show different intrinsic rhythms. Isolated myocardium from ventricles beat very slowly, while isolated atrial fibers beat very rapidly. Cardiac contractions are continuous and initiated by inherent mechanisms. Cardiac muscle fibers are long, cylindrical cells with one or sometimes two nuclei. The ends of the fibers split longitudinally into a small number of branches to give a continuous three-dimensional cytoplasmic network. To meet the high metabolic demand of strong continuous activity, extremely rich capillary network is present. This is supported by delicate connective tissue present between the muscle fibers. Like skeletal muscle, sarcomere is the contractile unit of the myocardial cell. It contains myosin and actin filaments along with troponin and tropomyosin. Shortening of sarcomeres causes contractions of muscles. Specialized intercellular junctions called intercalated discs are present between the ends of adjacent cardiac muscle cells. These are irregular transverse thickenings of the sarcolemma that contain structures called desmosomes. Desmosomes are like spot rivets. They hold adjacent cardiac muscle fibers together. The intercalated discs also act as points of anchorage for the contractile proteins and they contain important channels called gap junctions. These connect the cytoplasm of adjacent cardiac muscle fibers and allow the extremely rapid spread of action potentials from one cell to another. In this way, adjacent fibers are caused to contract almost simultaneously, which is very important if the heart is to function as an effective pump. Mitochondria are more numerous in cardiac muscle cells than in skeletal muscle. The numerous mitochondria, the large quantity of myoglobin and rich blood supply together make the heart very resistant to fatigue. All of these factors increase the rate of continuous aerobic metabolism. The T tubules are continuous with the cell membrane and these carry action potentials into the cell interior. They are well developed in the ventricles of the heart but poorly developed in both atria. The sarcoplasmic reticulum of cardiac cells is of small diameter and lies near the contractile elements. The function of the sarcoplasmic reticulum is the same as that of skeletal muscles, that is, they store calcium ions and release them for contractions. Three types of cardiomyocytes are found in the myocardium. Sinoatrial node or the pacemaker of the heart is a group of cells found at the junction of the great vein and aorta. These cells are autorhythmic cells and spontaneously discharge about 100 times per minute. Conduction fibers or Purkinje fibers allow fast conduction of action potentials around the heart. The most abundant cells in the myocardium are the contractile cells or myocytes. They are especially abundant in atrioventricular node. This node consists of small myocytes that conduct but delay the impulse from the pacemaker, thus allowing the atria to contract before the ventricles. No organism can stay alive without the functioning of the cardiac muscles. The ventricles or chambers of the heart stretch beyond their resting capacity when they are filled. They pump out the blood and carry it back to the heart. The cardiac muscle facilitates the pumping action of the ventricle. It squeezes blood out of the heart while contracting and retrieves blood by relaxing. It pumps the deoxygenated blood from the right atrium, a chamber of the heart, and right ventricle to the lungs, and pumps oxygenated blood through the left atrium and left ventricle to other parts of the body and the aorta. So, to conclude, we can see that muscles are the engine that our body uses to propel itself. It would be impossible for us to do anything without our muscles. Absolutely everything that we conceive of with our brain is expressed as muscular motion. Muscles are crucial to any animal and are incredibly sophisticated. They do everything from allowing us to walk to keep our blood flowing. They are efficient at turning fuel into motion, are long-lasting, self-healing and able to grow stronger with practice. So, we see how integral muscles are to our lives and how dependent we are on them for performing any action.